Good afternoon to everybody. Welcome back to our webinar in novel foods. Welcome to the afternoon section, which will be in bioactive ingredients and foods. My name is Márcio Carocho. I'm a researcher here at the Polytechnic Institute of Bragança, and it's with great pleasure that I will be introducing our plenary speaker, Dr. Daniel Granato, who is an associate professor in food science and health at the University of Limerick in Ireland. His background is in food engineering, and he has 2 PhDs, one in food chemistry and one in nutrition. He currently works with the recovery of bioactive compounds from food side streams, food authenticity, and bioactivity of functional foods. Today, he will be talking about functional foods, of course, but specifically the design of functional foods and the interface between innovation and practicality. So, Professor Daniel Granato, thank you very much for accepting our invitation. The stream is yours. Thank you very much.
Thank you very much. Let's see the technical checks now. Can you see my presentation?
Yes, perfectly. It's just shaking a little bit.
Yeah, I don't know what's that.
Maybe try and stop the presentation and start again.
Yep.
Okay.
Wait. Well,
it's still flickering.
Yeah.
Maybe adjusting the resolution of the screen.
Actually, it's not the resolution, so I will try here once more. Okay. I don't know what's going on.
It's still flickering.
Yeah.
And if you...
Daniel, are you sharing the screen or the application window?
Actually, I was sharing the PPT.
Try sharing the screen.
Okay, just a sec.
I don't know if it will help.
I will share the presentation again to see if it works. Nope.
Danielle, if you want, I can share it for you and then I
Yes.
will. Okay.
Okay. So can I send you? Or what do you suggest? Let me see if it works this way.
You sent it to me last time. If you want, I can present that one.
Yeah, I changed some
Okay.
things, but I think now you can see it maybe.
Yeah. Now.
Yeah, but it's still blinking.
Yeah.
I don't know why. It was perfectly okay.
Yeah.
So
just a minute.
yeah, I'm gonna send it to you.
Okay.
I know what it is. Wait. Now it's okay, I guess.
Yes.
Yeah. Okay.
So finally.
Sorry about that, but it happens. You know, that's the age.
Of course.
So guys, thanks a lot. So I would like to thank the organizing committee for having invited me to talk about a very important issue here, which is how to design a functional food and how actually we can assess any types of efficacy. And so I have heard and I have listened carefully, the talks in the morning and the ones to come. There are a lot of applications and how to create new potential functional ingredients and even foods. We do have lots of we do
Danielle,
have yes.
sorry, can you just turn your camera on, please?
Okay.
If it's not an issue with the presentation.
No, it's not. But I think the connection will not be good for my side, but that's okay.
Okay. Okay then.
Let's try it this way. I think now
Perfect.
it works, right?
Yeah.
Okay,
Yes.
okay. So today I'm going to talk about scientific definition and criteria, idea mining, and some expectations, efficacy testing. We're going to talk about translational SEGA, and then some concluding remarks. And I'm here to discuss ideas, and I'm going to give my point of view. Because I'm a reader of many different types of papers and different types of sciences. So I think starting off with the scientific definition and criteria, we can build some knowledge here, just like in these 35 minutes. So last year, actually there was a group that was actually trying to define functional foods in a way that, okay, so at this stage, the European Food Safety Authority, the FDA, they acknowledge that, okay, there might be some functionalities of some certain foods and ingredients, but how can we define functional foods? So this group decided to give a definition, and it would be natural or processed foods that contain some biologically active components, which in turn can be effective. They are non-toxic, and we need to prove clinically that they have some health benefits, and they can be used, to modulate, to prevent, manage, and treat chronic viral diseases and its symptoms. I have to make some notes about that. The European Food Safety Authority does not allow the words prevention, management, and treatment of diseases. So we cannot say that we are going to develop a food that will counteract viral disease or to control the symptoms of a disease, of a cardiovascular disease or cancer or such things. In food chemistry, journal Food Science, we receive quite a lot of papers that they are trying to convey like an idea that the functional foods they are actually trying to develop, they are good for viral infection or they are good for cancer, but it's not simple as that. In 2017, I decided to lead a group of people. So we had a chemist and we also had a professor in medicine. So we decided to look at the integrated approach. So when we consider chemistry, biology, nutrition, pharmacology, and also statistics for the development of functional foods. So that was a proposal, and that's what we actually believe in. So functional foods, they might have some potential positive effects on health beyond basic nutrition, helping the promotion of optimal health conditions and also reducing the risk of non-communicable diseases. So I'm not talking about treatment. I'm not talking about management. Okay, functional foods, they are not miracles. So that's what we need to have in mind. So it's not because we find that one food has antioxidant activity using certain methods such as DPPH or FRAP that these foods are functional. Okay, so I'm going to give you some of the criteria actually. If we look at the regulations, so just like a functional food, obviously it must be safe for consumption without medical supervision. So when we think about tea polyphenols, that in many countries, including the US, they are adding up to 1 gram of tea polyphenols in a capsule and saying that it's a nutraceutical. So EFSA took a look at that and said, well, more than 800, it can actually damage your liver. So we need to have some criteria, okay? When we are talking about food, and also when we are talking about nutraceuticals, okay? So the functional foods produce metabolic or physiologic beneficial effects through the action of a nutrient or a non-nutrient. So we can talk about protein, or we can talk about like a carotenoid. And obviously, it should be part of the daily eating habit. So which means that it should be chronic. It should be consumed regularly, right? And it must not be consumed as a capsule, a tablet, or a food supplement. So these are the most important criteria that we find in different regulations, especially in the EU. Okay, so the regulation 1924 from 2006, they mention everything about health claims. So what is actually good and what is actually approved by EFSA to have a health claim. So in Europe, the use of statements on health benefits, they should be regulated by the nutritional and health claim regulation. So this is the first thing. And why? Because these regulation, they actually protect consumers from misleading campaigns and also to harmonize the internal market. Okay, so this is something that it's really good about this regulation, because there is a harmonization about all types of requirements to provide a health claim. Is that easy? No, it's not. There are many steps to do that. And if we consider the combination between the academia and the industry, we might have to consider some other steps as well. And I'm gonna tackle all these issues right now. So by definition, functional foods have an additional effect or benefit that in practice, obviously, they are likely to carry an approved nutrition or health claim. So the regulation on nutrition and health ensure actually that these claims are appropriate, they are scientifically sound, and they are harmonized across European Union. So this is something really interesting. I'm just going to give you one example. The olive oil polyphenols. So we can add 5 milligrams of hydroxytyrosol into the olive oil. And if this quantity is present in at least 20 grams of the olive oil, it can give health benefits. There are clinical evidence to support the health claim. But just gonna show you one of the examples. So this is one of the papers that helped that hydroxytyrosol, which is this compound that you see there in olive oil. And the paper, one of the evidences that actually supported the allegation for the health claim of this functional olive oil. So as you can see, there is a randomized control trial to prove that the consumption of at least 5 milligrams of hydroxytyrosol in olive oil, and I'm talking about 20 grams, which we can do that very easily if we eat salads, for instance, or if we have a soup, that these compounds actually improve the endothelial function, especially when we are considering people with cardiovascular disease, for instance. Okay. But then, okay, we have some examples, but how actually a functional food is produced? So we need to have like an idea mining. We were talking about before about insects. We were talking about the cultivation of different species of macroalgae and so on as a source of protein, but how can we actually develop foods? So we need to consider the consumer. We need to consider economic factors, availability of those ingredients, and then obviously it's not something that we are going to do in the academia by ourselves. We can do some food models, but we cannot produce prototypes that will actually be absorbed by the industry right away. We need lots of tests, and especially if we think about the combination of the industry and the academia, can't we tell a story together to try to develop something together? Why not? We are talking about innovation. We are talking about IPs, intellectual properties. So our lives is not only about papers. We need to create impact, societal impact, and one of these things, why not trying to align with food companies at some point? So this is what I call the cloud concept. This is a modified concept from these authors, and I believe that when we are talking about functional foods, we have the food industry, they have needs. We have the food scientists that can deliver many different applications and tests and to produce evidence, right? But we also have social values and traditions. So when you go to Portugal, for instance, if you do not develop something with fish, uh, the people will say, "Well, come on, fish is super good. You know, everybody knows that." And then if you go to Finland, for instance, if you don't talk about berries, it's going to be a misconcept. So we do have tradition, and the consumer wants something that he knows already. It's easier for the uptake of the idea of a concept. But we also need to talk about technologies. We need to have technologies that are available. They are cheap at some point, and that can be used in many different places. So I can produce this food in the US, in China, in Africa, in South America, anywhere, or at least in a large part of place. And then we need to consider also some financial issues, right? Why? Because I can create a functional food that might be super expensive, but that's not actually the case. We can do something, obviously, functional foods will be more costly, but how much? So that's why we need the industry to help us out with that. I'm not an expert in finance, but I do tackle everything related to foods, right? And then from considering all the consumer expectations, the values, tradition, economy, the industrial needs, and what we actually can do to advance knowledge, so creation of new concepts, then we have the product innovation. But sometimes we are not an evolving cloud. We are an isolated cloud, which means that food industry is here, and then the researchers are here, and they don't combine. And then that's why it takes up to 20 years to develop a functional food. And to my opinion, these should be changed. And I think this is my personal opinion, and I'm trying to sell it in many different applications in the EU, that developing a functional food is like a storytelling. Why? If you see those errors, I am co-creating, taking into account the consumer, the nutritional value, the processing and prototyping, co-creation with the stakeholders, so with the industry as well. And I can go back at any time, because it's really important that we have these concepts all in one, because this is what we call storytelling. And I think the development of functional foods should be based on innovation, collaboration, and storytelling. So considering all those issues, there are many challenges, and some of them were already pinpointed by the other presenters. So this is what I think it's more appropriate to talk about right now. The price, taxes. In many cases, and I would say all the cases that we see worldwide, functional foods do not have any governmental incentives in terms of taxes. Okay, the collaboration between food companies and research institutions should be more effective. And that's not the case in South America. Here in Europe, we have this, it's a better way, but in many other places, it's not. Research investments coming from the industry are mandatory, but in some cases, they just want to give samples. And we don't need samples. Not only samples, we need money to buy stuff, to hire people, to buy reagents, to perform the analysis, right? And the regulation should be updated constantly. As I showed you, the regulation on functional food is from 2006. Then I ask you, nothing has changed maybe? Or we don't need to change that at all? We do need to change it, but we need to raise the flag and say, hey, this is what we need. So constant and research-oriented communication with consumers would be preferable. Now we have the internet. There are many ways, many venues to collect information from consumers, and that's something that I'm really interested in. But all those steps, they are required to deliver quality, efficacy, safety. So the consumer, money, and everything that we can do here in the academia. And then what should be done to develop functional foods with added bioactives? So I think that was the case from this conference, that we are talking about developing or creating or cultivating new ingredients that are full of bioactives, full of carotenoids, phenolics, and so on. But then I have lots of questions. Is the material normally used as food? Why do I ask that? Because I had like a student, he wanted to bring a herb from Indonesia, and that herb was not actually used as a food, and that herb was not approved in the EU. So we cannot move forward, even though it's reported that this plant has lots of bioactivities, or at least part of it. And then I start going into, I start moving into a more technical part of the presentation that I start. My questions are these compounds toxic? So maybe some of the compounds or the crude extract obtained with methanol, ethanol, and so many others can be actually cytotoxic to some of the human cells. Because if there is cytotoxicity, this plant cannot be used as a food. But if there is no indication, no evidence of cytotoxicity, then we can actually move forward and do some more tests. Okay, so that's why it's really important that we target the main compounds and perform some in silico studies to also predict the toxicology, the possible toxical effects of these compounds. And then we can move forward. I did in silico, which is modeling. Great. Now we can move into in vitro cell-based studies, toxicity evaluation, even in some cells in vitro. And then we go to the in vivo toxicity evaluation using a proper model. We can use flies, we can fruit flies, we can use fish, we can use many other types of animals as well. But then the efficacy testing in humans is mandatory, right? And that's what I called, this is a translational saga. So what is translational at the end? So how come, okay, I'm in the academia And my final destination in terms of delivering the results are the consumers. And I'm talking about functional foods. I'm talking about their health. So that's my main point. So I am analyzing the bioactivity of those compounds of those phenolic-rich extracts in cells, and how will this actually be functional? It's a series of different tests that should be done, as I was mentioning before. So when we consider our human cells, so we can use red blood cells, we can use white cells for that purpose, and then we can test bioactivity, we can test cytotoxicity of these extracts, okay, of these new potential foods, and then we can do some treatments with rats, for instance. In the EU, we try not to use animals for ethical reasons, and I'm going to give some inputs on that really shortly. And then when we see any results in rats, we can develop the mechanisms. We can understand and unveil the mechanisms of action. That's okay. We can try to assess if there is a dose dependency effect. And then, okay, everything is all right. Let's think about apple. Okay, apple, everybody eats apple. So I can do like a human intervention trial using humans. And I want to target, let's say, cholesterol levels, right? And then when I have in vivo data, especially considering this type of intervention, then these data can be used by EFSA to state recommendation on the use of certain foods or to approve a health claim. So it's not simple. Okay, so I'm just going to give you one example, something that we did with jabuticaba, which is called the Brazilian berry. So jabuticaba is this wonderful, super tasteful fruit. It's widely consumed, but the seeds are not. We just throw the seeds out. And then we optimized the extraction of these compounds, and the main compounds found in the seeds are viscalagine and castagine, as you can see in the table. So what we did, we were targeting antioxidant action and also anti-inflammatory action. So we did some in silico docking using different proteins like AKT1, SRS, and LRP3 inflammasome, and also MAPK1. And then what we did after seeing all those dockings, okay, let's try to do some validation of this in silico modeling using different cells and also using rats. So what we did exactly this time. We started with cells. So there was no cytotoxicity. So we went to the rats, and then we were analyzing that. We came to the conclusion that castagine and viscalagine are safe for human consumption, but that's okay because this is nothing new so far. And then we developed yogurt containing castagine and viscalagine coming from the seeds. And then we performed another study with rats. And then our main aim now is to proceed with a human intervention study and try to prove the efficacy, trying to validate this yogurt as a functional food. So this is the yogurt that we produced with different concentrations of jabuticaba seeds. And these are the initial observations that we had using different approaches. Okay, so the elagitannins from jabuticaba, they attenuated inflammation, oxidative stress, and also modulated gut microbiota in rats with cancer. So these results are available. We don't have right now a funding to go to a human intervention trial, but that's the focus later on. We even tried, okay, this is not the scope of our talk today, but we even developed some creams for the face using the jabuticaba seeds, and we also verified that the jabuticaba seed actually alleviates oxidative stress in human keratinocytes. So in different ways, we actually tackled and we proved that this is actually a potential functional ingredient. And then the translational research for functional food development is something like that. We have the basic research that we do in the academia. The clinical research can be done when we have more funds for that. And this is why working with food companies is essential at this stage. And we have the first translation from basic research to human studies. This might take up to 5 years, depending on the type of funding that you have, the type of research that you are doing from the basic science, and the amount of people or the type of approach that you're going to use in your human study. And then from clinical research to improved health, there is the translation of new knowledge into clinical practice. So I go to a physician and he's going to say, hey, that yogurt is functional, so it's approved by EFSA, and you should have like one a day. So this is the clinical practice that we are talking about. So as you see, there are many steps for that. And this is what we call saga. At least I call saga because it's not easy. It's not straightforward. Lots of money, lots of knowledge, and lots of input throughout many years. And obviously, it would be super interesting that the government would look at all this data and say, hey, we need to carry out a public health intervention into that. So we are going to create recommendations. We're going to create policies over those. And this is something that it's lacking in a more constant basis in the EU. So I'm just going to show you, okay, what is basic research? We start with in vitro studies. So chemical assays, enzymatic assays, cell-based assays. Why? Because they are cheap, they are super repeatable, they are easy to obtain. We can test toxicity, bioaccessibility, chemical modifications, but we cannot predict anything in vivo. So this is one bad thing. This is a very tracing assessment. And then I'm going to give you some insights on tissue engineering and why tissue engineering should be actually used more in food science and technology. Now we have started this year, actually last year, to build in vitro models of organs. And why is that important? This is widely used by the pharmaceutical industry, but we are going to use with food extracts, potential phenolic-rich extracts, carotenoid-rich extracts. We can actually build with these cells. They are commercially available. We don't have issues with ethics because they are just like we are not taking from humans, okay? Although they are from humans. So we can build them and we can see the effect of the extract on the liver, on the lungs, on the heart, on the gut, and we can have all those organs on a chip. So it's going to be like this size, 5, 10 centimeters. We can test everything together. So this is one way to counteract that. And then, obviously, to say that this food is actually functional, we need to go through a randomized, placebo-controlled, preferably triple-blind trial. So we have the healthy individuals or the placebo, and then we have the targeted population, and then we're going to do several analyses in the blood, in the feces. We're going to do some image testing. So there are many different types of analysis that can be done. And then I'm going to compare the populations using proper statistical analysis. And then I'm going to provide any recommendation. Is this food actually functional or not using this population? So overall, my to-do list is huge, because if I'm working with bioactive compounds, and many of you are, we have the source of these bioactive compounds, we have the extraction technologies, and then we're gonna make the chemical analysis and bioactivity. We are trying to develop new food models, and then we have to try to do some human intervention studies, so we can actually prove there is a cause and effect relationship between the consumption of this bioactive rich food and the health outcome. And this is the main aim. Only when this relationship is established is that I will talk about a functional food itself. Okay. So there are many examples in the literature and also on the shelves of the supermarkets that foods can be enriched with bioactive compounds. So even Activia is doing that in their shots. Okay, we have different actually edible flowers that can be used for such a purpose as well. As you can see here, this is the weekend. This is called Moscow Mule, and this is made of Clitoria ternatea. So it costs up to 50 euros in Asia, but this is a sensation, but it's made of a natural source. And then we can produce ice creams. We can produce cheeses. We can produce many different types of applications. Okay, using different herbs. Here you can see, I can send you this presentation for you later on. The amount of total phenolic compounds compared to the vanilla ice cream, it's like huge. I'm talking about the antioxidant activity is also huge in comparison with the vanilla one, which is 0, by the way. Incorporation of polyphenols in baked products, stability of natural pigments in wafers. So there are many and many countless applications, and this is a way to go. This is great, but we need to move further. But I also acknowledge that we need lots. We need money. We need incentives. We need projects. We need people to work with us to try to make it work, to make it functional at the end. Okay, so as a conclusion, I would say that bioactive compounds can be extracted from different sources and incorporated into foods. Different tests should be conducted, okay, to assure safety and bioactivity. In vitro, preclinical, animal-based, and also clinical trials are required. We need to establish the cause and effect relationship, and developing food products containing bioactive compounds should be integrative and very holistic. We cannot do that alone. And functional foods can only be regarded as such if there is this association between cause and effect. So it's a pity that I only have 35 minutes, but thank you very much for the attention. This is my email. If you want to contact me further on for projects and you want to discuss anything further, please let me know, and I'll be more than willing to discuss things further. Thank you.
Thank you very much, Daniel, for that enlightening presentation. We've got a couple of minutes for questions. I've got a question here by, actually 2 questions, one of them quite long, by Dr. Andrei Mokam. He asks, how do you see the involvement of medical personnel, namely nutritionists, gastroenterologists, pharmacists, in this kind of research? At the end, this will give a more confident use of these products by the patients or consumers. So he's asking the different personnel in this type of research.
As I was mentioning before, we need to be very integrative, and working with these professionals is essential. As long as that they are either in the academia or in the food companies at some point. But then when the product is already available, then the food company should actually approach these professionals and tell them, you know, why this product is special. And this has been done already in many cases, Andre. So this is super important. I really like your question because this is like, okay, afterwards, we need to communicate. We need to teach people why this is important. And one way to go is going to the nutritionists and to the physicians and say, hey, talk to your patient about that, because that might be something. So if we are talking about the functional foods, thumbs up.
Another question is, what, in your opinion, what is the constraint keeping the legislation from 2006 from changing? I've also read it. It's quite outdated. What would you say is the constraint of it, of EFSA and other entities not improving the legislation?
Well, it's very hard.
It's a tough one.
It's very hard, but I do think that the EFSA panel is there are not many people there as well. So I think there is also an issue of personal, and I think they are trying to be as strict as possible, because if they, you know, if they let many things just move forward, we know with this legislation and health claims, I think there might be some, let's say, problems in the market throughout the EU. We're talking about like many countries, so
yes.
So I think that's the one thing.
So I think we can go for one more question, also from Andre. Why do you say that in silico toxicology should be applied for bioactive compounds used in the design of functional foods? Will the producer use such a novel and unknown molecule to be needed to do this? This is normally made in pharmaceutical research on new molecules or new leading compounds.
Actually, in silico models are super easy to be conducted. And I think it's a lifesaver, to be honest, because if we have like a compounds or some compounds that can be targeted, and we are tackling, let's say, cardiovascular diseases or obesity or some types of cancer, there are lists of proteins that can be studied using these compounds. So instead of going to the lab, I can do this. I can do some modeling and see if there is any interaction. So I save time, I save money, I save resources. So although in silico models are used a lot in the pharmaceutical industry, in the food area, it's not. And that's why I'm raising my flag and say, we need to do better.
I also agree with you, Daniel. I mean, money and time, anything that can save money and time, it's worthwhile. So I don't think we have any more questions. We have to continue. So Daniel, thank you so much for the enlightening presentation.
Thank you. Thank you very much. Thank you very much, Marcio.
Thank you for the beautiful presentation. So now we will be going on to the next set of presentations. So I will be handing the stream and the session to my colleagues, Ileana Pereira and Cristina Caleja. From my part, thank you all for being here. We'll see you next time. Cristina, Ileana, please.
Thank you, Dr. Marcio. Good afternoon and welcome again to the webinar Novel Foods. We will start the bioactive ingredients in food communication section, which includes 4 oral presentations. Each speaker has 30 minutes to present, and to avoid delays in the program, please do not exceed the proposed time limit. I also remind all the participants who follow us on the YouTube channel that they will be able to leave their questions in the channel chat during all the section. The organization will select some questions that will be asked to the speakers at the end of the session. And for a matter of time, and it is not possible to explain all the questions, they will be sent particularly to the speakers to be answered later. The questions proposed by the channel participants, as I have already mentioned, will be exposed at the end of the 4 presentations. So we will start. I move on to the speaker, Giulia Bianchi, from Council for Agricultural Agricultural Research and Economics in Italy, to start her presentation entitled Environmental and Technological Factors Affecting Bioactive Compounds Content in Fruits and Vegetables. Giulia, you can start your presentation, please.
Thank you. Thank you for your presentation. I'm trying to start with the presentation, and I hope you can hear me and see correctly my screen. It's all okay, I think. Most bioactive phytochemicals belong to the secondary metabolism that is composed of several pathways that have their origin from primary metabolism products. Primary metabolism is common to all organisms, and it comprises the growth, development, and reproduction functions that determine the yield-related parameters. Secondary metabolism pathways have their origin from primary metabolism products and are often restricted to selected plant groups. They have a key role in the adaptation of plants to their environment, either for defense from predators or other biotic adversities. And these compounds have a relationship with a nutritional quality, while the compounds that determine the attraction towards dissemination of impollinators are related to the sensory quality. Since this topic is very important and complex, many authors have tried to summarize the existing knowledge, and many reviews are available, either general or restricted to a selected group of plants or a selected factor. I will describe in this presentation some example from the activity of our research center. In this study, it was performed a cultivation experiment with 14 hot pepper genotypes that were grown in 2 contrasting Italian locations. One in northern and one in southern Italy. This data refers to the variance, the analysis of variance, and total percentage total sum of squares indicates the percentage of variance due to each factors, and this indicated that genotype is the most was in this experiment the most important factor. But environment explained an important percentage of variation for carotenoids, ascorbic acid, and tocopherol. Capsaicinoids were an exception because the environment was not effective in determining changes. This probably is due to the fact that the genetic of capsaicinoids is quite simple. One single gene is responsible for pungency. So selection and breeding for this trait is quite simple. Another cultivation experiment on bell peppers was performed during 3 years at the certified organic experimental fields of the horticultural research of our organization in Monsalpolo del Tronto. 3 different cropping systems were tested, integrated, and 2 organics, one with artificial mulching and one with natural mulching. 2 genotypes were tested, 1 commercial hybrid and 1 local cultivar selected for organic production. The 3 years were quite different for weather conditions. 2060 was more rainy and less hot, while 2017 was characterized by a severe drought in summer. These factors were analyzed together and separately with a 3-way analysis of variance, which showed that for phytochemicals and antioxidant activity related parameters, the year was the most important factor. In particular, significant variation were observed for ascorbic acid, both spectrophotometric fraction of carotenoids, and the total reducing power and radical scavenging activities measured with Follin-Choqualteau index and DPPH. Some a significant effect was observed also for varieties. Regarding the volatile compounds profile, we can observe that cropping system was effective in determining the aldehydes and apocarotenoids content that were that were overall higher in organic crops, and in as regard the year effect, in 2017 were observed a higher level of 2-methoxy-3-isoputylpyrazine and methyl salicylate that are compounds related to the stress condition, to the stress reaction of plants. After harvest, the post-harvest management is essential to keep the nutritional properties of products. This experiment regards the mechanical damage and was performed on tomatoes harvested at the breakage stage and artificially damaged. After ripening and slicing, fresh cut tomatoes were analyzed for their lycopene content, and it was observed that mechanically damaged tomatoes have had a lower lycopene content. The same decrease on important nutrients can be induced by the technical operations like cutting for the preparation of fresh cut products. This example regards the actinidia fresh cut produce. Ascorbic acid was analyzed during storage, and it was observed that kiwi fruit slices had a lower content of ascorbic acid. This loss can be minimized choosing the proper storage temperature. One particular example of post-harvest management regards the reddening of a southern Italy apple cultivar, which is Annurca, and is a PGI product. After harvest, fruit is kept 2 weeks under a shadowy net on a straw bed, and this environment is called melaio. And fruit become red, and in this anthocyanin increase and polyphenol increase that is observed after this practice can be useful in limiting the post-harvest disorders such as superficial scald. The initial low oxygen stress can be effective in enhancing the glutathione content of fruit that keep the reducing form of ascorbic acid and ensures the capacity of the fruit to scavenge the reactive oxygen species. We can observe in this example that the glutathione content was sensibly higher in fruit that before the storage in a controlled atmosphere were subjected to 15 days in an atmosphere with a low oxygen content. Also, the choice of appropriate transformation parameters can be useful to minimize the losses in bioactive compound associated with processing. Thermal processing is known to have a detrimental effect on bioactive and on nutritional and sensory quality. In our institute, we performed a transformation experiments using mild technologies, which involve the use of lower temperatures. This case regards a miniaturized fruit transformation plant that operates at lower temperatures and under vacuum. With this plant, we can obtain a final products with the necessary value of Brix degrees. a transformation experiment was done with organic and conventionally grown strawberries, and the obtained product was compared to a homemade jam. The major differences regarded the better retention of ascorbic acid in jams made with both processes in organic strawberries, and the better retention of polyphenols in jams made with the mild technology plant. The impact of on sensory profile was different, because the mild technology involving the use of vacuum altered the volatile profiles, and the most esters that are the character compound in strawberries were reduced, but one of the most important compound, that is mesifuran, was retained in both types of jams. Another example of mild technology is the use of a solar dryer apparatus, which operated with day-night ranges of temperature and relative humidity. In this example, I show the results made that refer on dried peppers, and the effects on nutrient and volatile profiles content. Surprisingly, the reference product, which was a oven, which was a pepper dried in a conventional air circulating oven, were more rich in ascorbic acid, even if the transformation process involved a higher temperature, because probably the longer time was more was a principal effect.
Sorry, Julia, but you need to finish the presentation.
Yes, excuse me, I'm trying to finish. Another significant difference was the yellow carotenoids content higher in solar dry samples. Regarding the volatile profile, the oxidation products such as hexanal, which is an aldehyde, was higher in oven dry samples. Tetramethylpyrazine, that is an interesting compound that is absent in raw peppers, was found with high variable contents, and most in solar dried peppers, probably produced through microbial synthesis. and two important sensory-related compounds which is a terpenoid linalool and bell pepper pyrazine, were not affected by the drying process. So I finished my presentation. Thank you for your attention.
Thank you very much for your excellent presentation, Julia. We will move on to the next speaker, Miguel Angel Prieto from University of Vigo in Spain, with the presentation, Importance of the Optimization Procedures for the Extraction of Bioactive Ingredients and its application at the industrial level. Miguel, you can start your presentation, please.
I'm trying. Hello, Eliana. Hello, everyone, but it doesn't allow me to share my screen. Hello.
Julia has to stop sharing her screen. Julia, can you interrupt the presentation, please?
Julia, you need to finish your presentation, your screening.
Okay. Yes, I think now I can. Can you see my screen?
Yes.
So let me go back. I don't know why it's at the end. Sorry. Okay, can you see my screen, Cristina?
Yes, Miguel, you can start. Thank you.
Thank you, Cristina, for the presentation. And thank you for the invitation to this Novel Food Hub Systemic webinar. As Cristina said before, I'm just gonna try to present the importance of the optimization procedures for the extraction of bioactive ingredients and its application at the industrial level. My name is Miguel Angel Prieto Laje. I'm working in a group with other two important researchers as Professor Jesus Simal Gandara and Professor Jean Bourchau. We are located at the University of Vigo in the Faculty of Food Science and Technology. We also have strong relationships with Professor Lilian Barros from the Institute Polytechnic of Braganza and other researchers from Portugal like Professor Fatima Barroso, and from Argentina and Italy like Luigi Lucchini and Andrea Gomez. In our group, we are currently 17 PhD students and 6 postdoc students. Our main research lines are the chromatographic separation and extraction technologies applied for the characterization of secondary metabolites, bioactive compounds, testing their bioactive properties for the development of natural food products, and the valorization of byproducts and the application of bioinformatics and chemical engineering techniques in order to improve or quantify in a better way the response of biological properties and bioactive compounds. We use food matrices, byproducts, plant matrices in order to try to replace the synthetic compounds in a diverse range of products as functional ingredients or application in cosmetics or even to the pharmaceutical industry, taking always the focus of circular economic principles. The metabolites that we are just focused are basically phenolic compounds like phenolic acids, tannins, and flavonoids, or carotenoids, tocopherols, or unsaturated fatty acids. There is a wide range of compounds we are already focusing. The work process is we select a matrix, we apply a different or a set of extraction techniques, we identify and quantify the compounds that are interested for the industrial, we search for different biological properties, and we finally apply for a product. Then we can do this at scale up, but the problem reaches when we want to go to the industrial level, then is when we got the problem. Sometimes when we go to the industrial level, things are not easy as the scale-up process. So there is a pre-scale industrial level that has to be applied. And this is when optimization procedures are really need and welcome to help at the mean point between the industry and the research facilities. In the scaling up of process that has been established at the research facilities, there is involved several things like mathematical model, optimization, parameters, and also the outputs of the cost-efficient conditions that are helpful to the industrial level. When we consider a process that has been established in a laboratory facilities to scaling up, always there are different variables or large-scale volumes that are not easily to apply, or operational costs that were not initially taken into account, like minimum energy, time, and residuals. That this is not something that is new. This has been going on since the Egyptians. They trying to apply in Roman times as well for the wine production system, and today it's still happening. We are trying to optimize whether the use of solvents, whether the use of different conditions, whether the cost we get at the end of the process. This extraction process, when we want to optimize at the laboratory facilities, they are highly time-consuming process. If we want to optimize two variables like time and temperature, or power and solvent concentrations, the experiments can go up further than 100 experimental combinations, and these go up as we add different variables. Like if we want to optimize 3 or 4 variables, the experiments could go up to 200 or 300 experimental conditions, which are very unfeasible process and very costly at laboratory facilities. If we search in the Google search, for example, for optimization process, we get a highly amount of different results. Most of them are related with responsive phase methodology, which is a technique that can apply for different environments in order to optimize the responses. We have to take into account that this technique was developed by Box and Wilson in 1951, and this technique was applied for understanding how or how long different equipments last working with them in a daily basis, in a monthly basis, or in an hourly basis. These techniques are based in mathematical models as well as design experimental procedures that help researchers or companies to optimize different processes. Basically, it's based in a mathematical model, in a polynomial model with a second level with linear, interactive, and quadratic effects. It's able to reproduce or to describe the analytical responses of different variables from 2 to 10, and is able to describe how the process is going through. As well, we need an experimental plan. Also, Box were developed different experimental plans that allow researchers or industrial companies to apply different procedures. Currently, there are a massive amount of samples of different softwares that are able to help researchers or companies to apply their experimental designs to optimize a process. We can do it in Excel, we can do Minitab, MATLAB, or Design Expert. There are other available softwares. What we're going to present here is just an example we have done at the Scale Up Lab, and then it has bring it to a company. For example, in this case, it's just a simple case. It's the microwave-assisted extraction from brown algae for their step optimization for the obtaining a high GL concentration extract. In this case, we use 5 different algae, brown algae, and then we apply a microwave technique. We selected the same, at the same plant of 28 experiments, and we have Here, the variables that were used from time, power, and solvent concentration of water and ethanol, and these were the responses for the 5 algae species. Once this was applied with a mathematical equation, we got a set of parameters. Several of them were non-significant, and then we got an equation. For example, for this case of Ascophyllum nodosum, we got this equation. And also for the others, we got other equations. These equations can be used to bring up and to reduce the cost of companies in order to find those optimal conditions that can allow the process to be much simpler. For example, in this case, for an Ascophyllum nodosum, we can see in let me select here we can see how the conditions optimize around this point. The same happens for Bifurcaria and Bifurcata, and this all has been done with 20 experiments that can be mathematically modeled with a second-order polynomial model that allow us to understand the process. And easily we can understand the process is further supported by the solvent, then followed by the power, and then by the time used. So, for example, in this case, we were able to find out all the conditions that bring us a proper optimization process. This was scaled up to the industrial level, but those conditions might not work. That's why we are able to move around the different solutions that the optimization process allow us, and to decrease or increase the time and the power or the solvent concentration in order to produce a cost-efficient extract for the company with maximizing the extraction procedures. This also has been applied in several other cases. For example, here we have a case with the IPV, obtaining a natural compound, rosmaric acid, from Melissa officinalis, where we apply different techniques in order to understand which technique was much important or extract efficiently the rosmaric acid. In this case, we found out that the ultrasound acid extraction was the powerful technique for the production of optimized extraction. Also, the conditions were selected, and the process was patented in Portugal. In another case, for example, published in Food Chemistry in 2019, we also got a patent for the production of a natural colorant from Hibiscus sativira, petals extracted and applied to the industry. As well as the previous case, we have applied 2 different techniques like ultrasound and maceration, and we have tested the process at laboratory scale for making easily the description of the process to the transference to the industrial process. In this case, also, the better conditions were supported by ultrasounds, assisted extraction, and the process was patented. The same application, it can be done to different natural ingredients. In the case of Arbutus unedo, for also the extraction of natural colorants such as anthocyanins, in which also once again, in this case, the heat-assisted extraction produces the better concentrations and optimization extracts, and also was patented. As I said, these are processes that can be repeated constantly and optimized for transferring the results of an industrial level of the research level to an industrial level. Recently, another compound that has been optimized is the Chestnut flowers ultrasound-assisted extraction, which has a complex results because we studied all the compounds obtained in the extraction. So at the end, we got different flavonoids and different tannins. They were grouped together and they were studied together, and different complex results can be analyzed much further to obtain a better products for the industrial purposes. And so on, the results can be find out how the global conditions optimize the process. What I want to describe in this presentation was basically present a powerful tool that can be applied for the real growing demand of new natural ingredients for an efficient, cost-effective extraction method at the industrial level, and how these optimization approaches can be applied in a different ways from the researchers to the industrial, and how they can be feasible and attractive. They also have some involvements of mathematical models and solutions, but they are generally easy and accessible from the new software. And that is my presentation today. Thank you very much for your attention. And Eliana, if you have some questions, I'm welcome to hear them.
Thank you, Miguel, for your inspiring presentation. I'm reminding that the question for all the speakers will be asked at the end of the session. And now we will move on to the next speaker, Laila Meja from Riga Tarde University in Latvia. Laila?
Yeah.
So Miguel,
yes,
I'm closing. And Laila, you can start your presentation in the power of Rai, please.
I'm trying to close, sorry.
Yeah. It shows you cannot screen share while other participants sharing, please.
I'm trying, I don't know why.
Yeah, stop sharing.
Yeah, I'm doing it. So it's working? Yes.
Okay, Laila.
Yeah.
Can you add your presentation?
Yeah. Yeah. Okay, is it okay? Do you see me? Yeah.
Yes.
Yeah.
So you can start, please.
Okay. Yeah.
Thank you.
Good afternoon, dear audience. I'm a medical doctor with specialization in dietetic and nutrition, and my scientific interest is related to whole grain, especially grain fiber. Yeah, I feel quite inspired from some period of time from cereals to Magritte. This present period is also somehow serial, otherwise we would be sharing our knowledge in one room and later on we could have refreshing drink and could hug each other. But what is more, I have chosen this picture because of the title, The Cultivation of Ideas. And no matter what, I'm sure ideas are cultivating here. And nevertheless, I would like to express my pleasure about this webinar. Dietary guidelines around the world recommend people to choose more plant-based foods, and recommendations postulate eat more fruits and vegetables and choose whole grains instead of refined grain products, at least 3 whole grain portions per day. And it is considered and speak about whole grains when the ratio of bran, germ, and endosperm is the same as an original grain. In general, whole grains that are used daily in Latvia include wheat, rye, oat, barley, rice, and buckwheat. In addition to great nutrition value, grain products, especially bread, have also special historical and emotional value. And at the same time, there are so many alerts and frightening information in mass media. And it raises many questions. And firstly, eat or not to eat bread or grain products. And the evidence. And I would like to show this picture because it shows how exciting research is in the field of nutrition, and it is very relevant to today's event. Plant foods, including grain products, contain mixture of chemicals which are important in both prevention and treatment of diseases, and that therefore medical science is closely linked with food science because food functions as medicine. And clearly, evidence shows increasingly numerous health benefits of regular consumption of whole grain foods and a fiber-rich diet. Eating whole grains reduces risk of many chronic diseases like cardiovascular diseases, type 2 diabetes, and in addition, evidence shows that fiber-rich diet also lowers risk of many diseases, and even more impressive, fiber consumption is inversely associated with all-cause mortality. Looking at the structure, we find many protective substances, but special scientific attention is paid to dietary fiber complex and in Nordic countries to rye fiber complex, which is a mixture of biologically active substances like vitamins, minerals, insoluble and also soluble fiber, lignans, alkyl resorcinols. There are a lot of mechanisms behind mentioned protective effects like slower digestion, decreased hunger, increased satiety, and much more. And furthermore, we know that there are also so-called non-traditional cereals which have special features like hulless barley and hulless oat. And now about Latvia, what is happening in whole grain research, and I will give you some insight. For the first time, our scientists found lunazine peptide in triticale, and lunazine is a peptide with anti-cancer, antioxidant, anti-inflammatory, and cholesterol-reducing properties has been studied. And now about human studies. Here I have to tell some words about my PhD study, which was more in cancer area, but it was the start of whole grain research in human studies and also the start of research cooperation between medicine and agriculture universities. Some findings of our study. Knowing the importance of fiber in diet, it is important to identify fiber sources, and we found that the most important source of dietary fiber was grain products, 41% of total fiber, even more in males, which is similar to such countries as Finland, Poland, Denmark. If we look at grain fiber sources, rye bread played the most important role, accounting almost 60% of cereal fiber and 1/3 of total fiber intake, which is similar to Finland again. And importantly, very considerable substances in breads are alkyl resorcinols. They are found in outer parts of grains, moreover only in wheat, rye, and triticale, and in minor amounts in barley. Alkyl resorcinols are considered as potential markers of whole grain wheat and rye in grain products, and also as biomarkers for whole grain, rye, and wheat intake. They possess antioxidant activity as well. And when we decided to study the possible protective role of grain fiber, we realized that it is important to clarify the real amount of whole grain in diet in Latvia. For this purpose, we assessed alkyl resorcinol content in different breads. Lignans are other protective substances in cereals closely related to fiber intake in countries where main lignan sources are cereals. And lignans are main phytoestrogen source in Western diet. We assessed lignan content in most popular Latvian and also Finnish breads. In addition, we have studied lignan intake in main food sources of lignans in Latvian men and women. And the major food source was bread, seed and rye bread. And we were investigating alkyl resorcinols metabolites in prostate cancer patients, and results suggested impaired metabolism in prostate cancer patients. Next, about other projects in area of grain research. This study covers many steps from breeding crops, evaluating the dietary value of a new variety of hulless oat, then producing breakfast cereals, and finally clinical intervention study which includes patients with inflammatory bowel disease and controls. Majority accepted taste of oat flakes, and more than 1/3 reported subjective improvement of symptoms after oat consumption, and concentration of antioxidants is increased. And the results suggested that hulless oats could be part of daily diet due to acceptable taste and gastrointestinal symptoms reducing properties. And other study, in order to evaluate the health potential of innovative grain products, glycemic and insulin index in different grain products was assessed. We investigated germinated and non-germinated products. Here we can see in the investigation of glycemic and insulinemic response of barley and activated barley flakes. And these results were interesting and promising. Flakes from activated barley show lower glycemic response and more clearly very low insulin response. Moreover, when we analyzed flakes from different activated grains, barley, triticale, and rye, and all show low glycemic response, but barley the lowest, whereas rye the most stable concentration of glucose, prolonged blood glucose response, which is clinically very important. And what is more, we studied postprandial glycemic and insulinemic response after consumption of activated wheat and triticale grain flakes. Activated triticale flakes induced lower insulin response and more stable concentration of insulin, whereas activated wheat flakes induced only improved glycemic but not insulin profile. The beneficial effects could be associated with multiple compounds such as insoluble and soluble fiber, antioxidants, anti-inflammatory substances, and the mode of action is supposed to be synergetic, including slower digestion rate and the effects of the gut fermentation processes. And result of the study was generation of new flakes for breakfast with potentially protective effects against metabolic syndrome. So, and coming back to diet recommendation and food choice, we see quite relevant for us Nordic nutrition recommendations, which are based on local products of our region, and fiber comes from whole grains with dry bread in the first place, and next to barley, oat, and others. And our Latvian recommendations say that the recommended fiber intake is 35 grams per day, which is, you know, quite higher in comparison with other countries. And it is known that in the past, whole grains were a part of folklore and culture, and Finnish proverb is rye in the wrists, and our Latvian rye empowered both the man and the horse work hard, but nowadays it's scientifically proved that dietary fiber complex has protective properties, like it's a Weight Watchers, food protects heart, improves gut health, and many other effects, and inspire of both scientists and manufacturers for new healthy product concepts. Yeah, my supervisor, very respectable Professor Herman Adlerkreutz from Helsinki University, liked to show this picture of Marc Chagall during the First World War. Rye bread helps the soldiers to survive. But nowadays, our whole grains help us to keep fit, to give us energy, health, and beauty. And before COVID, in company, but today more lonely, but still with strength and drive to endure, enjoy life. Thank you. I finished.
Thank you, Laila, for your excellent presentation. So I remind all the participants who follow us on the YouTube channel that can continue to post their question in the chat to be exposed to the speakers in the final, okay? So let's move on to the last presentation. I present Michaela Verni from Università degli Studi di Bari in Italy. Michaela, you still
Hi, good afternoon. Yes.
You can start your presentation, please.
Sure. You should see it now. This presentation is entitled Reposing Brewer's Spent Grain, Tailored Bioprocessing to Improve its Antioxidant Properties. So you can start.
Good afternoon, everyone, and thank you for the introduction. So 39 million tons is the amount of brewery spent grain generated every year, which, to give you an idea of the enormous volume we're talking about, it's enough to fill over 1 million shipping containers. So here's a reminder of why this process, we should find a solution to this process. This process is not sustainable. Currently, the majority of spent grain is used as low-value animal feed, whereas a little part of it is used to produce biogas or dispose of the landfill. So imagine not only the cost of disposing of such huge amount of spent grain, but also the environmental impact of moving this much highly perishable byproduct. Yes, because 80% of it is made of water. But if you look closely, you'll see its potential, since the remaining 20% is made of fiber and proteins, and it's also rich in phenolic compounds. And this begs the question, why isn't brewery spent grain used as food ingredient? Well, its use is limited by its complex structure and the poor technological performances. So treatments are necessary to reduce the negative impact on both texture and sensory properties of the final product. And this was basically the aim of the FAMBREW project, improving the nutritional and technological performances of spent grain through bioprocessing technologies. So after screening of 33 lactic acid bacteria strains, we selected 3 Lactobacillus plantarum based on the ability to improve the in vitro antioxidant property of spent grain. Then we set up a protocol involving an enzymatic treatment with a commercial silanase and fermentation with selected starters. Bioprocessed spent grain had radical scavenging activity towards DPPH and DBTS, as well as the ability to inhibit lipid peroxidation, which was comparable to that of alpha butyrhydroxytoluene. But however, since in vitro assays can only be considered as predictive tool of the antioxidant activity in vivo, and testing substances directly on human or animals is not an easy approach, ex vivo assays offer the right compromise to better understand food functional properties. So spent grain extracts were tested on human keratinocytes subjected to oxidative stress. And we observed a protective effect of the bioprocessed spent grain, which was comparable to that of alpha tocopherol. So we decided to study what changes happened in the matrix that made these changes. And of course, the first thing we studied was the phenolic compounds. And spent grain, as well as many other cereals, is mostly made of free phenolics and those bound to the lignocellulosic material. In our case, most of the effect was because of the enzyme, the xylanase, which released most of the phenolic acids bounded to the cell wall materials, ferulic acid derivative most, and degraded the Arabic oxidant structure, releasing many other compounds. But lactic acid bacteria also enabled the release of the release of other phenolic compounds. More specifically, lactic acid bacteria metabolized phenolic acid into more active forms through a series of decarboxylase and reductases. And since not only methanolic extract had antioxidant activity, but also water-soluble extract, we hypothesized a contribution of substances other than phenolic compounds. In fact, the proteolytic system of lactic acid bacteria enabled the release of small bioactive peptides, which are sequences encrypted in native barley and mice proteins, which have sequences really close to those of antioxidant peptides. And these peptides were separated and identified to mass chromatography coupled to mass spectrometries. So once we established the main changes that happened during bioprocessing, we went back to what was the aim of the FAMBREW project, which was not only recycling spent grain, but also making it a valuable functional ingredient for food applications, which is why we use spent grain by process according to the protocol I described before as a 15% replacement of semolina to produce a fortified pasta. And of course, this pasta could be labeled as high fiber and source of proteins according to the current European regulations. And compared to semolina pasta, but also to pasta containing just native spent grain, by-processed spent grain pasta had higher protein digestibility and higher quality indexes, as well as lower glycemic index. But most of all, by-processing improved the technological properties of pasta. In fact, the degradation of the arabic structure resulted in the release of compounds which were entrapped in the cellular compartment and in a more homogeneous distribution of protein, spent grain protein within the gluten network. And the extensive cell wall degradation also enabled the release of compound which confer in vitro mimic digestion of the pasta. The digested pasta showed protective effect towards human colon carcinoma cells. In fact, when the cacao cells were incubated in the presence of by-processed pasta, they had the lowest range of reactive oxygen species. Now, the fortified pasta showed a peculiar sensory profile with cereal, malt, and sourdough bread-like taste, and also a little bit of bitter taste, which was, however, markedly improved by the pre-treatment. And this leaves us with one last question, which is, are consumers willing to buy spent grain-containing products? And the answers appear to be yes, because at the University of Helsinki, they conducted a little study which revealed that consumers are not only interested in spent grain containing products, but they also want to be involved in the product development. And they see spent grain a good fit not only for products like bread and pasta, but also for products like granola bars or cookies and crackers. So to conclude, by increasing the shelf life and the functionality and technological performances of spent grain, a highly perishable byproduct was was valorized by processing technologies, and the addition of processed spent grain in food formulas can increase the intake of fibers and proteins, as suggested by the Food and Agriculture Organization, can also increase the intake of active compounds. All of this while fulfilling consumer needs for healthier food and fostering cooperation among several players in the supply chain. Now, I would like to thank all the people that worked at the FAMBREW project, the brewers that provided the spent grain, and of course all of you for listening. And I will leave you with a little take-home message, which is the proper bread processing technologies can get us a step closer to unleashing spent grain potential in a more sustainable food system. Thank you.
So thank you very much, Micaela, and thanks to all the speakers for the brilliant words and the presentations. And now we will start the question session of participants who follow us through the YouTube channel. Let me check if we have questions. Yes. I have one question for Micaela.
Yes.
Did you try fermentation with fungi instead of or in combination with fungi, which could probably be quite efficient in liberating potent precursors? Could you give us an idea of sensorial notes of BSG?
Okay, no, we didn't try fermentation with fungi. We only tried it with lactic acid bacteria, and since lactic acid bacteria don't possess all the enzyme necessary to degrade the structure, we use commercial enzymes. And the idea about this, the taste, it doesn't taste like beer, but it has this like malt scent. But anyway, in pasta, when it's mixed with semolina, it's you can really taste that much of the spent grain depending on the quantity of addiction.
Okay, excellent. So very little late, so we only make one more question for Dr. Miguel Prieto. Dr. Miguel, what is the relation of cost benefits of your work, of this optimization? Dr. Miguel? Okay. So then we contact Dr. Miguel. So thank you again to the speakers and also the channel participants. Now we will continue the session of oral communications with the moderation of Dr. Filipe Reis and Dr. Oludemi Tofik. Thank you all.
Okay, okay, you are listening to me. So good afternoon, everyone, and welcome to this last panel of presentations of the first webinar on novel foods based on new ingredients and materials and process, sorry. So let's start this last panel. And I remind the speakers that they have about 13 minutes to make their presentations, so that in the end we have 2 minutes for the audience to ask some questions. The audience, as you know, can send their questions through the YouTube channel, chat, sorry, and we will address these questions to the speaker. So now I will introduce our first speaker, Ksenia Nikolkova from the Riga Stradins University, that will present her work entitled Antioxidant and Anti-Radical Properties of Pomegranate Juice. Ksenia?
Yes.
Are you with us?
Yes, just a moment.
We can hear you.
Sorry, do you hear me?
Sorry, Ksenia, we can hear you. Maybe you have the microphone off.
No, we can hear her. Yes, everything's okay.
Yes? Sorry, it was mine. Sorry.
No, it's
so,
okay.
Welcome, Ksenia, and the floor is yours. You can start your presentation. I'm so sorry.
Mm-hmm.
Yes.
Yes, just a moment. Yes. Yeah. Everything is okay. So good afternoon one more time. Thank you for the invitation to this webinar. My name is Ksenia, and today I'm going to speak about, so my research topic is antioxidant and anti-radical properties of pomegranate juice. So, as we know, oxidative stress is an imbalance of oxidant production and antioxidant responses when the former exceeds the latter. And its consistent components are active forms of oxygen and nitrogen, as well as other free radicals. And oxidative stress has a significant impact on the onset and course of several diseases because free radicals can adversely affect various classes of biological molecules, such as proteins, lipids, and nucleic acids. And it can lead to the problems in cardiovascular, respiratory systems. It can also lead to the neurological disorders and other complications that you see from the slide. And Today, much attention is paid to the foods, the consumption of which would contribute to the improvement in preventing diseases and also slowing down the progression of existing disease. And the most well-studied products that are rich in antioxidants are, of course, wine, it's coffee and tea, it's also oils different, especially if we are speaking about olive oil, and also fruits and vegetables. And although the consumption of plant products in the concept of healthy and balanced diet is associated with health benefits, many people still consume them inadequately. And one more possible source of antioxidants is pomegranate juice. And it could be a good addition to the body's internal antioxidant potency, mainly hydrophilic antioxidants. And as you see, it has different chemical compounds like phenolic acids, anthocyanins, flavonoids, and so on. And due to the very wide spectrum of different antioxidants, they also provide a wide spectrum of action on free radicals. And regular consumption of pomegranate juice can also lower oxidative stress, reduce it. It exhibits antibacterial, anti-cancer, anti-diabetic, hypertensive, hepatoprotective, and other properties. But there is one thing that we should take into account, that the composition of this juice depends on many factors. Thus, for example, environmental factors, variety, type of storage, processing conditions, and so on. It has there was many studies where it has been reported changes during processing in antioxidant amount and also in antioxidant activities. And those, the aim of our research was determination of total amount of antioxidant, also evaluation of the ability to reduce potential free radicals. And this was performed analyzing both freshly squeezed pomegranate juice and also industrially produced juice. So the research was performed in the Latvian, the Biochemistry Scientific Laboratory of Riga Stradiņš University. And for analysis, we took five pomegranates from different regions that were available in Latvian retail. So these pomegranates were from Spain, Turkey, Azerbaijan, South Africa, and Egypt. And also we analyzed 4 industrially produced pomegranate juices from Azerbaijan, Turkey, Georgia, and Ukraine. And for determination of total phenolic compounds, we use phenolical reagent. Then to determine the anti-radical capacity, we use the PPH method, and the ability of antioxidant to reduce the trivalent iron ion was evaluated using FRAP method. And comparing all results, we saw that these results were different. So the highest total content of phenolic compounds was found in industrially produced pomegranate juice from Turkey and Georgia, as well as in manually squeezed juice from South African pomegranates. And freshly squeezed pomegranate juice from Egypt and also Azerbaijan showed the lowest value in phenol compounds in this reaction with phenolical reagent. Then speaking about antioxidant activities, so about the ability of antioxidant to bind the PPH radical, the highest values were found for industrially produced juices from Turkey and Azerbaijan. Slightly lower it was for manually squeezed juice from South African pomegranates and Georgian industrially produced juice. And the lowest antioxidant activity, as it was previously, was found in Azerbaijan pomegranate juice. And the highest ability to reduce trivalent iron ions to divalent iron ions was determined to industrially produced pomegranate juices from Turkey, Georgia, and Ukraine. And you see also that among all juices, higher values are industrially produced juice. In the practical part of the study, it was also possible to compare freshly squeezed juice and industrially produced juice from one country of origin. So it was in case of Turkey and Azerbaijan. And despite the fact that processing could partially destroy biological active compounds, we saw that results in all three methods were higher for industrially produced juices. So to conclude, we saw that these results were different for pomegranate juice from pomegranates from different regions, and also for pomegranate juices from different countries of origin, if we are speaking about industrially produced juices. And higher values in all three methods were obtained for juices from South African, Turkish, and Spanish pomegranates, and commercially available juices from Turkey, Georgia, and Azerbaijan. And one possible explanation is that during industrial production, it's possible to extract biological active compounds from all parts of pomegranates, so from inner membranes, from peel, and from seeds also, not only from areoles. And there is also possible practical use of the results because, as I said previously, the products that are rich in antioxidants could be a good addition to the body's internal antioxidant potency. So it's all for me, and thank you for your attention.
Thank you so much, Sinia, for your great presentation and for the time that you spent presenting that. I'm just checking if we have some questions from the audience. We don't have any yet. I had one, but you almost answered it in your conclusions that were
explanation. Yes.
Yeah, the explanation why you obtained these results. The industrial extraction was effective. Since you, like you said, you could extract the compounds from different parts of the fruit.
Yes, because they are rich in antioxidants. Yeah, that's it.
Yeah, okay. So, and you were expecting that? Because usually when we are working with natural products, we just associate the natural one as the most effective, comparing with that is processed.
Results from different studies that were available for this moment when I started my research, there were different. Some of them say that there's a decrease in these antioxidants, and some of them or about enhancement in this antioxidant ability. Yeah, so there was different, and I was not expecting this.
Yeah. Great. I don't know if you want to continue with this work and perform maybe some other assays to prove what you find with these first chemical assays. If you have some plans to
I
continue.
had an idea, yes, about this. Yes.
Okay. So thank you so much for your presentation.
Thank you for the invitation.
Thank you. And I will pass the speech for my colleague, Taufik.
Thank you so much, Filipa. I will introduce the second speaker, Ana Rita Silva. She's a researcher from the Mountain Research Center here in Bragança, in Portugal. And she will present her work titled Cytinocytosis and its great bioactive potential. So this is just a gentle reminder that if you have any question and comments, you can just put them in the YouTube server, in the chat section, and we will send them to the speaker accordingly. Ana Rita, you can share your screen and your presentation.
So, I'm trying to share my screen, but I'm having some issues. I don't know why. Just a second. For some reason, I'm not capable. Sorry. I have it open.
Ana Rita? Do you have two screens?
No, I don't have. I just have one, and I'm trying to select. I select the presentation, and when I press, it does not allow me to share. I don't know why. Maybe it's better that you follow to the next presenter, and I will try to sort it out. I'm really sorry for this inconvenience.
If you want, you can send me the presentation, Ana Rita, and I can show it on the screen.
Okay, I will send you. Oh.
So while Ana Rita sends me the presentation, Filipa or Tofik, can you please ask Francisca to start her presentation, because she says she's also ready to do it now.
So since Francisca is ready, I think she can start. So I can introduce her. Welcome, Francisca, and thank you for being ready. So let me just check. So Francisca is our next speaker since we are waiting for Ana Rita. She's from Requinte, here from Portugal, and she will talk about a product, or rather the residues of a product that is very familiar to us here in Bragança, the chestnut. Francisca, welcome again. You can start our presentation with the title Chestnut Husks as a New Potential Active Ingredient, an exploratory approach with eco-friendly techniques.
Thank you very much for your kindly presentation. It is a pleasure to be here today. Good afternoon, everyone. My name is Francisca Rodrigues and I'm from Requinte, Porto. I'm located at the Superior Institute of Engineering of Porto. And today I will present you our most recent work entitled Chestnut Shells as New Potential Active Ingredient, an exploratory approach employing eco-friendly techniques. And first of all, I would like to thank this opportunity to share our work and interests with all of you. So, the idea of this project started due to the high amounts of food byproducts. The problematic behind this study is based on this huge amount of food wastes that is generated across food chain. According to the most recent data, almost 1/3 of the world food production is lost, being estimated that just in Europe, this value may reach 90 million tons with associated costs of 143 billion euros. The principal causes for these losses occur not only at farm level, but also at industry, supermarket, and even at home. So the challenge is to sustainably achieve new ways to valorize these wastes, aiming to obtain their bioactive compounds by green extraction techniques and principles, and use them as new ingredients for food and cosmetics, for example. So at this point, in my opinion, it is imperative to distinguish between food losses, for example, fruits without caliber to be commercialized or damaged fruits, and food byproducts such as shells that are considered wastes and should be removed from the production line. And our work is focused on a food byproduct, on chestnut shells, aiming to reuse them and add value to these byproducts. As it is well known for everyone, Castanea sativa is a chestnut species widely disseminated in Europe. The fruit that is represented in this figure is very appreciated in southern countries such as Portugal, Spain, France, or even Italy, being also used, for example, for gourmet food such as marron glacé. So during chestnut production, different byproducts are generated, such as leaves, for example, and even flowers that are traditionally used in medicine by infusions and decoctions, as well as burrs and shells. And shells can be considered the principal food byproduct generated. Almost 50,000 tons of chestnut were produced in Portugal in 2021. And just to have an idea, during chestnut processing, almost 10% are inner shells and 13% are outer shells. So with this project, we decided to add value to these extracts, and the idea behind the use of extracts rich in antioxidants from natural sources in cosmetic products, for example, is to combat the oxidative stress. External factors such as UV rays or even pollution may lead to the generation of pro-oxidant reactive species, oxygen species and nitrogen species. We know that the human body is characterized by an antioxidant defense capacity that is composed by enzymatic and non-enzymatic substances. The imbalance between the antioxidant defense capacity and the pro-oxidant reactive species may lead to the generation of oxidative stress, a phenomenon that is responsible for damage not only in DNA and proteins and lipids, for example. And this may lead to skin damage. And at the final step, this skin damage may lead to photoaging and even photocarcinogenicity. So our idea is to use these natural extracts obtained using green extraction techniques to scavenging this reactive species and stop the oxidative stress. Our studies and the studies of other research teams allow us to conclude that chestnut shells are rich in polyphenols, particularly phenolic acids and tannins, but also vitamin E and amino acids. And its richness in natural antioxidants have attracted the interest of food, cosmetic, and nutraceutical industries. And besides that, chestnut shells extracts have present different skin effects. For example, protective effect against inflammatory and UV-induced DNA damages, sunscreen's effects, the capacity to improve the collagen synthesis, the osmotic balance, and the skin hydration. And for example, and we already observed this in our in vivo study that is now being finished, the decrease of the wrinkles amplitude and skin roughness. So this is a really good new metrics to be used. As shown in this diagram, I decided to summarize our study. We start by dehydrating the shells that were supplied by a local company. We milled the shells and then we extracted the shells using different extraction techniques, namely ultrasound-assisted extraction, subcritical water extraction, and supercritical fluid extraction. For all these techniques, the optimal extraction was optimized using a mathematical model, namely response surface methodology, in order to achieve the best time and temperature of extraction. So after determining the optimal extraction binomial, the temperature and time, the optimal extract was characterized regarding radical scavenge capacity, cell viability, using not only skin cell lines, but also intestinal cell lines, 2D and 3D models. The phenolic profile was also studied, as well as the permeation using Franz cells coupled to human skin, the elastase and dehydrogenase inhibition, and other biological activities. So the optimal extract was then selected to be tested for a nutraceutical application and the cosmetic application. Today, I will talk about our results regarding the cosmetic application. The optimal extract was then tested in vivo on human volunteers in a patch test and incorporated in a cosmetic formulation. And this cosmetic formulation was also developed considering a mathematical model. The other part of the project included in vivo assays in mice and the development of a functional product. So our studies reported the richness of the free extracts, all of them, in phenolic compounds with a particular emphasis on ellagic acid, catechin, epicatechin, epigallocatechin, and caffeic acid derivatives. We also evaluated the radical scavenge capacity, and particularly the subcritical water extract and the ultrasound-assisted extract achieved the best results in these studies. Considering that, we start with in vitro assays. So fibroblasts and keratinocytes that are skin cell lines were exposed to the different extracts in concentrations ranging between 0.1 and 1000 micrograms per milliliter. And as shown in this table, for the subcritical water extract, the extract did not affect the fibroblast's viability for all tested concentrations, while in keratinocytes, just the highest concentration led to a decrease of the viability to 57%. The results are similar when we exposure our cells to the ultrasound-assisted extract, and the worst extract that led to a low viability was the supercritical fluid extract. Regarding the elastase and the hyaluronidase inhibition and enzymes that are present in skin and associated with skin aging, the best results were also achieved by the ultrasound-assisted extract and the subcritical water extract. Considering the phenolic profiles and the previous results, and always taking into account the European regulation of cosmetic ingredients and products, we selected the subcritical water extract to be tested on 3D skin and ocular models. We have here a 3D skin model, the EpiSkin, and the Skinetic human corneal epithelium that is a validated ocular model. So after exposure to the extracts and controls, it was possible to conclude that the subcritical water extract did not decrease the cell viability in both models, as it is possible to observe, being in line with the negative control used that was PBS. Oppositely, the positive control that was sodium lauryl sulfate led to a viability of 0 in both models and a high release of interleukin-1 alpha. And these results allow us to conclude that the extract was classified as non-irritant for skin and eye application. After we decided to perform an ex vivo assay to evaluate the compound's permeation, and we performed this assay in France cells coupled to human skin. We have a protocol with the local hospital, so we decided to use the human skin. And after 8 hours of assay, it was possible to quantify the principal phenolic compounds, particularly after exposure to the subcritical water extract. And we were able to quantify not only ellagic acid, but also epicatechin, epigallocatechin, and catechin. Based on that, we performed a patch test in 20 volunteers. And for that, the extract, as well as the positive and negative control, were in contact with skin during 24 hours by a patch test, as shown in this figure. And after that time, the patch was removed and the edema and the erythema were evaluated. And the results revealed that the extract was slightly irritant just in few volunteers, and probably these results were a consequence of a patch allergy to the adhesive observed, but no edema was observed in any one of the volunteers. So at this point, we could say that our extract was safe for a final cosmetic application. So finally, we develop a cosmetic formulation with the extract as aqueous phase. We also use it again, a mathematical model, because we want to achieve a pleasure and stable formulation. We performed the scale up of the formulation, and the final formulation was studied in stability studies during 3 months at 40 degrees and 75% of relative humidity. And we can prove that our formulation presents the same characteristics than at time 0. So we are now performing the anti-wrinkles assays. They will finish at the end of February. And the first results at time 28 were really interesting. At this point, we know that we have 3 different extracts that are rich in antioxidants, particularly the ultrasound-assisted extract and the subcritical water extract. The in vitro results and the ex vivo assay proved the permeation of the phenolic compounds. And we also demonstrated the safety of the subcritical water extract via patch test. So we hope soon we have the products already prepared to go to the market. So finally, I would like to thank to all these people and institutions that helped me in the successful execution of this project. Thank you very much.
Thank you. Thank you so much, Francisca, for your super interesting presentation. And congratulations on this new work designed to develop a new cosmetic ingredient extracted from chestnut shells and with proven bioactivity and absence of unwanted effects. I'm just checking if someone has a question or any doubt. I guess not. I have just a curiosity. It's not a very a question so far, but a curiosity. Since you use different greener extraction techniques like subcritical water extraction, as you said. Can you tell me, do you have good extraction yields that will allow you to produce higher quantities of this ingredient? And do you think that these ways of obtaining the extract are viable for its production in an industrial scale that I hope you can reach?
Thank you very much, Philippa, for your questions. Yes, we have very good yields. For us, 20%, 25% is really good. It's really good. And we already tested at the extraction in huge amounts at a semi-level industrial, industrial level, and the results were really interesting. So we trust soon we have a new extract on the market. Yes.
Okay, yeah. I'm expecting for it to try.
Thank you very much.
Thank you, Francisca. So if we don't have more, yes, we have one question. Joanna is asking, were the chestnut extract tested in non-allergic volunteers, or were any allergic patients included in the assay?
No, for the in vivo assays, just volunteers without any kind of allergies can be selected. But this is a good question, because we know that sometimes people are allergic to some compounds that are used in cosmetic extracts. And without that, this could be a new study to be performed. Yeah, it's really interesting.
Yeah, but you are following all the legislation. So
yes.
Okay, so good luck, Francisca, with your work, and thank you again for your presentation. So I guess we can move on to the next speaker.
So I don't know if Rita is ready for her intervention.
I am ready. I'm still having technical issues, so Lilian is gonna share my presentation.
Okay, perfect. So you can start. The floor is yours.
Okay. First of all, I would like to thank the opportunity to present my work. My name is Ana Silva. I'm a PhD student, and today I will be presenting the most relevant results regarding my PhD work. Today's presentation is titled Cytinus hypocystis and its great bioactive potential. Lilian, please. Thank you. So Cytinus hypocystis is a wild edible parasitic plant, and according to the few studies published in literature, it was used in the past in folk medicine to treat several health conditions. Therefore, and to unveil the potential of this unexploited species, we established two main objectives: the chemical characterization of Cytinus hypocystis and the determination of its bioactive properties. Next. The first step to chemically characterize Cytinus hypocystis was to evaluate its nutritional value. We performed a comparative study between the whole plant and its nectar. Regarding the results, we understood that both samples presented similar energy, ash, and carbohydrate content. Unsaturated fatty acids prevailed over polyunsaturated fatty acids. Fructose, glucose, and sucrose were the soluble sugars identified, and oxalic, malic, and citric, the organic acids. Protein and fat were twofold higher in the nectar when compared to the whole plant. Next. We also evaluate the phytochemical profile of the whole plant and its three main parts, namely stalks, nectar, and petals. We tentatively identify 16 hydrolysable tannins and 1 catechin, and because the phenolic profile of all samples were identical, I will be only presenting the most relevant results regarding bioactivities of the whole plant. Next. To evaluate antioxidant activity of our extracts, we used two methods, auxilia and tubars. Auxilia evaluates the capacity of the extracts to retard oxidative-induced hemolysis in sheep erythrocytes, while tubars evaluates the capacity of the extract to retard lipid peroxidation. In both assays, our extracts exhibited a lower EC50 than the positive control, Trolox, which is a potent antioxidant, synthetic antioxidant. Next. To evaluate the cytotoxic activity of our extracts, we used the colorimetric sulfurodamide B assay, 4 tumor cell lines and 2 non-tumor cell lines. Cytinus hypocystis extract was able to inhibit the proliferation of the 4 tumor cell lines, and no toxicity was observed for the non-tumor cell lines up to a concentration of 400 micrograms per ml. Next. The pinitrophenol colorimetric assay was used to evaluate the in vitro antidiabetic activity of our extract using yeast alpha glucosidase. During this assay, the CH extract exhibited a lower EC50 than acarbose, which is a known medicine used to reduce glycemia. Next. So during the previous work, we were able to establish a correlation between the bioactivities and the tannin content in our plant. Therefore, we performed an extraction optimization using ultrasound-assisted and heat-assisted extraction to maximize the concentration of tannins. RSM was applied to find the optimal combination between variables to maximize the final response. The best extraction methodology was ultrasound, since high temperatures during heat led to compound degradation, and also ultrasound offered advantages in time and energy consumption. Therefore, the following bioactivities I performed using the optimum extracts from extract optimization. Next. So to further analyze the anti-diabetic activity of my extracts, we established a collaboration with our colleagues from University of Maringá in Brazil. There, they evaluated the capacity of our extracts to inhibit several enzymes involved in carbohydrate digestion. Contrarily to yeast alpha glucosidase from rat was less inhibited by our extracts, but we have an excellent result for human salivary alpha amylase and porcine pancreatic alpha amylase. Following these results, our colleagues performed an in vivo assay using healthy and diabetic animals. During this assay, CH extract reduced blood glucose after starch injection in both models. Next. Tannins are also promising compounds to be used in the cosmetic industry. Therefore, in the next 3 slides, I will be presenting related assays with this field. Tyrosinase is known for its main role in melanin synthesis. Therefore, finding tyrosinase inhibitors to mediate skin hyperpigmentation is a hot topic in the industry. To evaluate the anti-tyrosinase activity of our extracts, we used L-DOPA as a substrate, MBTH as a chromogen, which binds to the oxidation product dopaquinone, forming a stable complex and allowing a more accurate determination. During this assay, our extract exhibited a lower EC50 than kojic acid, which is a well-known commercialized inhibitor of tyrosinase. Next, please. For the wound scratch healing activity, an artificial gap was performed in monolayers of keratinocytes. After the scratching, the cells were incubated with citinous extracts, and we were able to see that after incubation, the untreated cells had a wound closure of 26%, while the cells treated with our extract had a wound closure of 67%. Next, please. Finally, we also evaluate the antibacterial activity against skin pathogenic bacteria. Citinous extracts exhibited activity against the 4 tested strains, being its most effective action against Staphylococcus ludonensis. Therefore, we also perform an adhesion and invasion assay to determine if our extract were capable of protecting ACAT cells from Staphylococcus ludonensis. As we can see from the graph, our extract were more capable of inhibiting invasion than adhesion. Next. Concluding, Citinus hypocystis is a hydrolysable tannin-rich plant exhibiting multiple bioactive properties, and considering the previous results, our focus will be on its cosmeceutical application due to its properties, namely antioxidant, anti-tyrosinase, wound healing, and antibacterial. Next. Thank you for your attention.
Thank you so much, Ana Rita. Thank you for your interesting talk. Let me check if we have any question for you on the server. No, until now. So just to ask if you have any desired interest in the host of this plant, because as a parasitic plant, of course, there's a host. What are you going to do going forward with the host of this plant? Is there any interest?
We already performed a comparative study between the bioactivities and the phytochemical profile of the host and the parasite. And although the host has also some interest, we were able to understand that the parasite was more effective for the bioactivities we selected. So although we study and release the information to the public, our main focus is with the parasite, and I will continue with this work to finalize my doctoral degree.
Okay, great. Let me see if we have any more questions. There's still time. So until now, nothing. So thank you so much, Ana Rita.
Thank you. Sorry for the technical problems.
No problem at all. If there's any question, we will send them to you via email. Thank you so much for your intervention. And we will move now to the next talk. I will introduce the last speaker of this section and the last speaker of the webinar, Thomas Skok. I don't know if he's online. He's from the Technical University of Munich, and he will present the work titled Fiber Substitution in Common Foods and their impact in human metabolism. Dr. Thomas, if you are ready, the floor is yours.
Oh, thank you very much. I think you can hear me, you can see me.
Yes, we can hear you perfect.
Very good. Okay, thank you very much to give me the presentation to present our interesting data on some convenience products which we developed during a federal ministry-funded nutrition cluster. My name is Thomas Kurt, and I'm heading the core facility for human studies at the Thiel Institute for Food and Health at the Technical University of Munich. Today, I want to present our nutrition cluster called Enable Healthy Food Choices in All Stages of Life. One work package of this highly interdisciplinary cluster is named the Valorizing Fiber Intervention Studies, which I will present the next minutes. The mission of our cluster was to develop a healthier and tasty convenience food product or more products for all stages of life, particularly for groups at increased risk of dietary related diseases. You can see at the bottom which we defined those groups at risk, vulnerable phases of life, 4 groups shown on the bottom here: children, young adults, adults, and seniors. As I said, it's a strong interdisciplinary approach, including various academic partners and various global players of industry. And we want to contribute to the development of sustainable food with a high acceptance in the population. If you are interested in some data of the cluster and the project I'm sharing with you today, you can refer to this publication, which shows you the design and all the data which are available within this cluster. One particular aspect of this cluster and of our project is dietary fiber and the health benefits. We know for many years that there are protective effects on the development of coronary heart disease, stroke, hypertension, diabetes, whatsoever, and some types of cancer. Increased fiber intake enhances also weight loss in obese individuals. Increased intake of soluble fibers and insoluble fibers improves glycemia and insulin sensitivity in type 2 diabetes. And what is interesting to note is that the common recommendation for the intake of fibers is 30 grams. And nowadays, in mid-Europe, we only reach 20 to 22 grams. So it's far beyond the official recommendation. Fibers are difficult, and they are various fibers. They can be either soluble or insoluble. They can have varying water binding capacities. The viscosity is changing. Absorption capacity is higher or lower, and the electrical charge can be different. So there are various types of fibers which can be used in technology to produce new types of convenience products. In all our studies, we have a deep characterization and phenotypization of our volunteers. We use scales, ultrasound, blood parameters. As you can see, we have indirect calorimetry. Sometimes we use biopsies as well, and many, many questionnaires. For this study, we invited people for 2 entire days to donate their data for our cluster. The fiber intervention trial, it's more interesting to read it in German because it's the fiber Ballaststoff intervention, it's abbreviated FBI, aims to investigate consumer acceptance of various fiber-enriched foods, as well as the effects on metabolic and cardiovascular risk factors. There are 2 studies, a pilot study with 2 common convenience products with the substitution of 20 grams of fibers, and a long-term intervention study for 12 weeks, which aims to increase the intake of fibers by 10 grams. The first and the second intervention is using 2 commonly used food products. And I can tell you this food product on the left side, the Bavarian Leberkäse, is really endemic and it's highly consumed in Bavaria. I think pizza is more a global player, and it's the top seller if it comes to salami. In our study, we were able to produce it in a wild type and as a placebo product, and then together with fibers. And you can see the fiber-enriched meal contained 19 grams of fibers, the standard meal 2.6. Similar, in the pizza, you have 20 grams of fibers compared to 6 grams in the control pizza. From the aspect, you cannot find any difference between the products, so we were able to do a double-blind placebo-controlled intervention. It's striking to see that the fat content, in the fiber-rich meal is substantially lower than in the standard meal, which is reflected in the end by a 30% lower energy content of the fiber-enriched meal compared to the standard meal. As this was an acute intervention, I only summarize some aspects, which are glucose and insulin. As you can see, there is expectedly not that much difference between both types of food, the wild type and the fiber-enriched food. However, although the effects are not dramatic, but if you consider a 60% reduction in fat and a 30% reduction in energy, one could imagine in the long-term run that there is a substantial effect on body weight. Coming to the fiber acceptance study, this is the 12-week intervention. We took a basket of various fiber supplemented foods. Again, similar in the aspect, similar in the texture, exact the same in the taste. We used a middle age group of 40 to 65 years, and 110 people took part in this study. It was a 12-week study, as I mentioned, and they have 3 visitations at baseline, after 4, and after 12 weeks. Here you can see what types of food we offered to our volunteers. This included bread, rolls, pretzels, muffins, chocolate cake, lemon cake, leberkäse, fruit desserts, mousse au chocolat, vegetarian pizza, pizza salami, soups, different soups with noodles, without noodles, and various mueslis containing different amounts of fibers. People could select from this basket the food items freely over a time for 12 weeks. And in the end, we could compare wild type foods with the fiber enriched foods. To show that it principally worked, we see that the intervention group, which contained 77 volunteers, substantially increased their fiber intake, as we wish, by around 10 grams. So this was effective in the end. But what about the metabolic effects? If we look to all participants from the intervention group, and I only refer to the intervention group as there was nearly no effect in the control group, we can see that in cholesterol and LDL cholesterol, there is substantially a reduction of the blood levels between visitation 1 and 3. So between the beginning and the end, we have a reduction of cholesterol LDL, and the same is true for insulin and the HOMA index. That means in all our volunteers, they profited from an increased intake of only, to say, 10 grams of fiber per day over a 12 weeks period. We could do a subgroup analysis, as this group contained around 50% of people with risk for metabolic syndrome, which we defined with an elevated waist circumference of 102 centimeters in males and 88 centimeters in females. What we can see, the effects are similar, but slightly less effective in this group, which started basically at higher levels. However, also those people profited, especially here in insulin levels in the HOMA insulin resistance index, which was higher for those people with the metabolic syndrome compared to those lean volunteers in our group. Of course, we took a look at the metabolome, but fair enough to say, after 12 weeks, there are only minor changes with only a few really bacteria showing differences in their abundance, which might be related to the sequencing of 16S of the bacteria, which might not be the adequate model to show differences in the microbiota profile. In summary, I can say fibers can be used to reduce energy content in convenience products, which might also facilitate the reduction of meat for the production of food. Technological processes can provide fiber-enriched products with identical appearance to the wild-type foods I mentioned. As there are different fiber types, we have many new technological options to produce reformulated products which have a high acceptance. And I did not mention the acceptance here, but we measured this, and this was recently published from our group to see that the products having the same appearance, having the same texture, having the same taste are of course accepted at the same level, although they might be a little bit more expensive. And people are willing to pay this. I have to confess that we have a very selected population, as it is always if we perform intervention studies in nutrition. In most cases, people take part who are really interested in nutrition, and therefore these results, of course, might be biased a little bit. Long-term application of fibers reduce the cardiometabolic risk factors, which I have shown for total cholesterol, LDL cholesterol, insulin, and the HOMA index. And this is what I mentioned, the consumer acceptance towards such product is high, but has to take into account eating motives and the attitude of the individuals. Please give me a last remark, as I'm guest editing an issue of nutrients named Precision or Personalized Nutrition, which is now open for submission. If you are interested to contribute, please come back to me. Then you can be able to receive special conditions. The deadline of this special issue will be May the 25th. Now I want to thank you for your attention, and hopefully after this presentation you are hungry. Thank you very much.
Thank you so much, Dr. Thomas Koch, for your interesting intervention. We have one question here. It's from PB. It says GOS or FOS, are they considered in your studies?
Sorry, the what? Yeah.
It says that GOS or FOS, are they considered in your studies?
GOS or FOS, what does that mean? Sorry.
If GOS or FOS are considered in your study?
No, this is what we did not take into account yet. So we performed many analyses, but not all. And I would have to look which data are still present, and maybe we can take a look at this in a later analysis, but not yet.
Okay, thank you so much. I think we don't have any question until now. Let me see if we have any. Nothing. We don't have any question. Thank you so much, Dr. Thomas, for your intervention. And we've come to the end of the section. I want to thank all the speakers for their exciting presentation, and thank everybody for following us on the YouTube server as well. I hope you all enjoyed the webinar, and we will hopefully meet very soon, presentially in other conferences as well. We are running out of time. I will now pass the floor to the chairs of this webinar, the organizing committee, Dr. Lydian Barosh and Dr. Pascal Banan. Thank you so much.
Thank you so much, Philippe and Taufik, for their for the chairs of this last session. And now we are almost finishing our first webinar on novel foods. It's almost concluding and on time, even though we did have some technical problems. But it's normal because when we are online and with distance to everybody, sometimes it's difficult to overcome some situations. So I would like to thank you all, first of all, for your patience when these problems actually did occur. So for me and for Pascal, it was a great honor to organize this webinar. And actually it was the first time I organized a webinar with Pascal from IRE. And it was an honor for me to also do this, organization with you, Pascal. In our opinion, because I've been speaking with Pascal, it was a very successful webinar. So therefore, on behalf of CIMU and INRA, we would like to extend our most sincere gratitude to both the plenary speakers that were here with us today, in which we had two of them, one in the morning, Dr. Luca, one in the afternoon, and Dr. Daniel, but also to all the speakers that were here throughout the day, in which we had 16 oral communications, 8 in the morning and 8 in the afternoon. But we also want to thank all the assistants that we had through our YouTube channel while we were streaming. We had a great level of participations, and we want to thank you all for joining your efforts to be with us during this journey in science, which is very important. We are also proud of the numbers that we obtained for this webinar. As already mentioned this morning, we had around 400 registrations for this webinar. And what I have seen from the YouTube video of this morning, we already have around 420 visualizations. So this transmits the success of this webinar. Just want to give this note that we have developed a little booklet with the abstracts that will be on the web page homepage, where you can download afterwards the PDF with all the summaries of the oral communications that were performed during today. Pascal and I would also like to take this moment to thank the organizing committee, Taufik, Bruno, Jean-Marie, for their collaboration in this organization of the conference, though we must give a special thanks to Jean-Marie, who was with us in all the stages of this webinar. We would also like to thank all the chairs of the sections today, Sandrina, Angela, José, Inês, Carla, Márcio, Iliana, Cristina, Filipa, and Taufik, for following all the presentations and adding such interesting questions to our speakers. To the Innovation and Web Project Center, especially to Ricardo, for his help on the online streaming. As all of you know, these last 2 years have been a great challenge for all of us that are involved in science, and 2022 continues to show some of these challenges. Although, in the words of Carl Sagan, which I know quote, "Science is not perfect. It can be misused. It is only a tool, but it is by far the best tool we have, self-correcting, ongoing, and applicable to everything. So therefore, we need to keep improving this tool in our specific areas in order to overcome all the current challenges that we are facing. So for now, this is what I have to say to you. I want to thank you all. I want you to pass some time to Pascal, for I'm sure he wants to say some.
No, nothing to add. Your conclusion is really perfect. So I nothing to add. No, it was really a good moment, a good, and what people don't see always, it's the preparation behind this and all the people involved in this organization. So it's really a good experience, human experience, and I hope we will have some others in the future.
This is our first novel food.
Yes, so we will make
another webinar.
How many per year? I don't know. We'll see. We'll discuss on this.
So thank you all. I want to wish you all a very nice evening and a good weekend to all of you. So yes. Goodbye and